Welcome to These Kids Can't Wait, discovering the world of rare disease. Welcome back to These Kids Can't Wait, discovering the world of rare disease. I'm your host, Erin, joined by my co-host, Laurel, and today we're honored to interview a true pioneer in the world of rare disease treatment. Dr. Paul Orchard has spent decades pushing the boundaries of what's possible for patients with lysosomal disorders.
Dr. Paul Orchard is the medical director of the Inherited Metabolic and Storage Disease Transplantation Program and a professor in the Department of Pediatrics in the Division of Pediatric Bone and Marrow Transplantation at the University of Minnesota. His work includes blood stem cell transplantation, gene therapy, research into enhancing enzyme delivery, modifying stem cell transplant approaches, and the development of combination therapies to improve patient outcomes. Over the course of his distinguished career, he has become a leader in the world of rare disease, especially lysosomal disorders. Dr. Orchard's groundbreaking work has helped shape treatment strategies that offer real hope to patients and families living with conditions once considered untreatable. Dr. Orchard, thank you so much for joining us. To start, can you share a little bit about yourself?
Sure. Yeah, I'm not sure I can live up to all that as an introduction. I trained in pediatrics, University of Wisconsin in Madison, and then have come here to Minnesota and did some additional training in hematology, oncology, and blood and marrow stem cell transplant. So I've really pretty much been here since that time. So it's been a few decades at least. And then over the course of my career, I did less and less the oncology stuff, so I don't really do that anymore. The kids that take care of leukemia is a kind of a different group, for instance. And then I've been more interested in the rare disease population. And so probably over the last 25 years or so, really, I've been focused on the use of stem cell transplantation for those types of disorders. So really at this point, those are the kids that I see in my clinic. Those are the families that I talk to and interact with.
What first drew your attention to lysosomal disorders?
Well, the University of Minnesota kind of got on the ground floor of looking at transplantation as an option for these types of diseases. So the first lysosomal child that was transplanted in the US was done here in 81, I think. And so that group was up and running and were funded and are already seeing a number of patients. And a lot of families were coming here for evaluations and therapy. So that group already existed. Bill Crivet, who was one of the pioneers in that, was here. And so I started going to those meetings and then became more and more involved. And we lost Bill about 20 years ago. And so I've kind of taken that on. And so that's kind of how I see my role, is trying to better understand these diseases, what therapies we have available, what new things might be coming down the pike, and how we might better treat the kids.
Yeah. Looking back, what were some of the biggest challenges in those early years when you started working with ultra rare conditions?
Well, there's been a lot of modifications in the transplant procedures over those decades. So initially when we started, if you didn't have a brother or sister who was a good match, you couldn't get a transplant, essentially. And so back in the late 80s and moving forward from there, we've had more options in terms of being able to find a donor. So for instance, there's about, I think, 30 million people now that have been tissue typed, and the National Marrow Donor Program, or the NMDP, has a database of all those people that have said that they might be considered as a donor. And so we've got a lot of different donors to look at. And then the cord blood route became available 20 plus years ago too. So there's a lot of blood stem cells in placental blood. And so as people identified that and realized that maybe this could be used for transplant too, there's been a number of centers that have been creating banks of cord blood, essentially, or placental blood. And so those are available as possible uses for transplant too, for to serve as donors. And so those types of things have made a huge difference. The morbidity and mortality of transplant is still a challenge, but a lot of that has improved kind of dramatically as we've got better donor choices available. We've been able to fine tune the chemotherapy a bit too, to try to back off on that as much as we can safely. So initial protocols that we used were designed for the kids that had leukemia because we were familiar with those and we knew those worked. But in those patients, obviously you're trying to eliminate the leukemia population over and above just get the donor cells established in the person being transplanted. So we didn't have to use regimens that were quite as intense as we did for the kids with leukemia. So determining how much we can back off essentially and make the kids less sick, but still get the donor cells to engraft is the term that we use. It was really one of the things we've been refining over the last 10, 15 years as well. So we've modified our current protocol a number of times and are still kind of doing so, but it's gotten a lot better. And as you're well aware, I mean, we still make kids pretty sick and they're in the hospital for a long time. And it's a difficult process for the families to go through, but it's not the way it was 10, 15 years ago.
I'm just curious. So when you're talking about, cord blood versus a donor, do you have a preference? Does it matter?
Well, there's pluses and minuses of both, essentially. So the cord bloods are somewhat limited by the number of cells that you have available. So they get as much blood as you can out of a placenta. So, essentially what happens is the moms are going to deliver in an institution where they're doing cord blood banking. They will ask if it would be okay to use the placenta and to extract the cord blood and potentially use that for someone that might need it. So they get as much blood as they can out of the placenta and they count the number of cells there, but it's, usually around 100 mLs or so. And so there's a finite number of cells there, really. So that's one of the limitations on the cord blood is you can only get as many cells as you can extract from a placenta, essentially. And the number of cells that you give on a per kilo basis makes a difference. So the more cells you give, generally, the better things tend to work. So that's one of the limitations on the cord blood side. The other one is there's only so many cells there, and if it looks like the patient's slow to have their counts recover afterwards, you can't go back to that donor, which was a baby that may have donated 10 years ago. You can't go back to that baby and get some more cells. So then you'd have to go to a different cord blood and kind of start all over in terms of who your donor is going to be.
Have you had to do that before?
Yeah, I mean, occasionally that happens where the first, there's an infection or something, and the initial infusion of cells don't take really, don't grow. And if you in a situation where you don't have any immune system for an extended period of time, as you can kind of imagine, that increases the risks. So we don't want to have someone devoid of an immune system for any longer than they need to. So if it looks like it's taking a real long time for the cells to come in, we would consider giving another infusion. But again, you'd have to go to a different cord blood unit.
Yeah.
One of the things that's helpful about the cord bloods, though, is they're already there and they're banked and they're in liquid nitrogen and they're ready to be used. So if we identify a cord blood that we think might work well for a particular patient, we double check the tissue typing and make sure that they're a good match. But after that, you're pretty much ready to go. You just order the cord blood and you can move forward. So that's quicker than having to go to unrelated donor. So the unrelated donor thing is, so these people might be anywhere within the US or even internationally. And we would generally pick the best donor. But when we identify a donor and that donor might be in St. Louis or might be in Berlin, and they would come in and have an evaluation done, make sure that they're healthy, and because we don't want to put them at risk if they have to go through a bone marrow harvest procedure and go to the operating room, because these people generally are doing this out of the goodness of their heart, really. So we don't want to hurt the donors, so we have to make sure that they're healthy enough to go through the process, and we want to make sure that the donor doesn't have some sort of issue that might influence how the patient being transplanted is doing. So donors have to come in, have an evaluation done. There's blood work, maybe chest X-ray, and maybe some other things that they might need to have done. And then we have to get all that information back, and then we would set up a time for the harvest. But you have to kind of work around the donor's schedule on these things, because again, they're doing this on their own, and if they have to take time off of work or whatever, then we have to arrange it on that basis. So that can take substantially more time than having to just use a cord blood, for instance. But if you need to go back to an unrelated donor because you want to get more cells, because things are coming in slowly, for instance, you have the opportunity to do that. The donors aren't obligated to provide additional, cells, but most of them become invested and would like to see that things work out well. So generally speaking, you can go back to the donors and get some more cells. So anyway, those are kind of the considerations that we tend to think about. But when we are looking at somebody that might need a transplant, we have to get their tissue typing or HLA typing to help us find the donor. But we generally search both the cord blood and unrelated donor sides simultaneously, and then help decide what we think is the best choice for whatever patient needs a transplant.
Yeah, there's a lot that goes into it for sure. Looking back, can you remember what it was like to transplant the first child that you ever transplanted?
Well, so I was involved actually in some of the transplants back when I was doing some training, like back in Madison. And there was a patient there that had another really rare disease, osteopetrosis, essentially, is what that little girl had. And we ended up, again, we didn't have a good donor for her, so we ended up using a kind of a mismatched donor. And she went through transplant, and this is a skeletal disease, essentially. And she was doing well from the standpoint of recovering afterwards and whatnot. And so the people there were saying, well, we should break this up. And I was a second-year resident. I said, well, I'll do it. So we wrote that paper, and then people started calling me about these diseases and saying, we saw your paper, you're the expert on this. I was like, well, no, I'm not really the expert on this, but we are interested in these types of diseases. So and that's one of the reasons I actually chose to come to Minnesota, because Minnesota had such a strong transplant program. And that's why I've stayed.
Yeah. Where are you from originally?
Originally from North Dakota, actually. So I went to school there, got initially into medical school there, transferred out to Brown. So I went out to Rhode Island for a few years, and then again came back to Madison, and then back to Minneapolis.
Okay, yeah. So you're kind of close to Montana. Do you ever go back to North Dakota?
I still had some family there, so yeah, I'm from Bismarck initially, so
Nice.
Went back and forth there, obviously a number of times, but don't make it back as often as I used to.
Well, we love Minnesota. Big fans of Minnesota. It's a great state.
Yeah, it's been a nice place to live and a nice place to work, and so there's a lot of really interesting things going on, and so we're looking forward to what's going to be available over the next 5 to 10 years too, because I think it's really an exciting time.
Dr. Orchard, for our listeners who are not familiar, can you explain how bone marrow transplants work as a treatment for lysosome disorders?
Yeah, I mean, the basis of this was set way back when. So there was a paper that was published in 1968 where they had the Liz Neufeld's lab was the group that was working on this, but they had cells in culture from patients that had lysosomal disorders. This was Hurler syndrome and Hunter syndrome at that point. And you could show that those cells, the lysosomes become very large and swell, and the cells look quite abnormal. And the story goes that somebody who was culturing the cells ended up mixing them together. And at that point, they didn't know that these were, with what enzymes were involved or any of those types of things. But essentially what they found out is that if you put normal cells in some same cell culture, the same Petri plate, essentially, with cells that are derived from a patient that have lysosomal disease, that the lysosomes get better, and that accumulation of the substrate, whatever the enzymes are supposed to break down, kind of disappears. And so that the normal cells can correct the issues associated with the cells that are from a patient that has a lysosomal disorder. So they termed that cross correction. I mean, that was how it was kind of billed. And so as people were talking about this, they said, well, we're figuring out how to do bone marrow transplants in people. If we could take cells, a bone marrow, for instance, from somebody that doesn't have one of these diseases, and establish them in somebody who has an enzyme deficiency, that those cells potentially are going to circulate through the body for the life of the patient, essentially, and serve as a source of enzyme. And so that was the rationale, really. The first transplant was actually done, was using the mother of a patient that had Hurler syndrome in Bristol in the UK in 1978, actually. So it goes back kind of that far. And then, as I mentioned, the first one in the US was done here, but that was really sort of the background. So this was before all the genes were identified and cloned, and now it's relatively straightforward to take one of those genes and introduce it into a cell and have those cells make enzyme that you can then bottle, essentially, pharmaceutically, and be able to give to people. So the enzyme replacement therapy for the lysosomal disorders came on 15 years or so after we started doing transplants. So transplants was all we had for an extended period of time, really.
Mm-hmm. And how do you determine whether a patient's a good candidate for a transplant? Are there specific criteria that you look for?
Well, so there's a couple of answers to that, I think. So the bigger picture one is how confident are we that it's going to help a disease? There's a lot of lysosomal storage disorders. There's 50, 60 lysosomal disorders, and transplant may work reasonably well for some and not very well or hardly at all for others. And so that's one major, distinction in that for individual, and you guys both know this well, right? So can you extrapolate the experience with some of these other diseases that, these are all rare, but some of the ones that for which there's more experience, and say that that's going to be applicable to another disease? And the short answer to that is no. We just don't. We can't really say that because it works for disease A, it's going to work for disease B. So that's one whole set of questions. And within a given disease, some kids are sicker than others, and some kids have more rapidly progressive disease than others. So there's a lot of variation even within a particular disease about where the kids are. So it's easier to make an impact if the kid's disease isn't as severe, and it's easier to make an impact if you're treating them very early in the course of the disease as opposed to later. It's overcoming problems that are associated with a particular disease is harder than helping prevent those from happening, if that makes sense.
Yeah. So you really have a window.
Identifying the kids as early as you can and intervening as early as you can makes a huge difference. And so I mean, there's been a lot of interest in the newborn screening route for some of these diseases and whatnot, to kind of on that basis, not just in particular for transplant, but for some of the other interventions that are in existence too. And some of the diseases have clinical manifestations that can end up being important. Some of the lysosomal disorders, and it can affect the heart and the skeleton and the airway and all these other types of things too. And if the kids are sicker from that standpoint, taking the transplant can be, well, that increases the risk of transplant. So if you've
it
got,
has to be hard on their body.
well, that's exactly right. So, I mean, it's a very stressful thing to take someone through. And if your kidneys aren't working very well, your liver isn't working very well, or you've got lung disease, or you've got, other types of problems, or cardiac disease, that increases the risk of going through the transplant procedure too. So we have a kind of standard package of things that we look at to try to establish whether a patient has one of these types of problems, because we want to know about that before we take them to transplant, rather than find out about it as they're going through the transplant process.
Are there different types of variants within a disease that respond or don't respond as well to a bone marrow transplant?
Yeah, I mean, so for instance, we do a lot of transplants in kids that have Hurler's syndrome. So Hurler's syndrome it's an alpha L iduronidase is the name of the enzyme. But there's a lot of variation in the severity of it. So the kids that have severe disease tend to have a lot of skeletal problems. They have eye issues, airway issues, cardiac issues, all these other types of things. But they also have neurologic issues. And if you want to get into this kind of now, but I mean, I think one of the reasons we still use transplant for these diseases is because the enzymes that are available, and again, all these genes have been cloned, and so all these genes are can be used to make a pharmaceutical product. But those pharmaceutical-based enzymes are very large molecules, so they don't penetrate into the brain very well. And so the neurologic diseases are not very well treated with enzyme therapy. There are some cells that are from the donor that after transplant can make their way into the central nervous system and serve as a source of enzyme. The number of cells that get in is pretty limited. It's actually pretty hard to know how many cells we're actually delivering, because you can't tell by imaging or anything like that. And you'd have to do.
I was going to ask if you've ever done a biopsy. Like, have you ever had that tissue to study from the brain to see what's making it in?
It's very limited. There was a group actually relatively recently that published the experience, not in the lysosomal patients so much, but the way this study was done is that women that had gone through transplant and had died after, and this wasn't done here, but they'd gone through transplant, died, they were transplanted with male cells, and they had an autopsy done. And then you can look in the brain to see what proportion of the cells are male, essentially. There's a test that you could do to try to figure that out. And so again, there's a variation in terms of what people are being transplanted for, what they died from, what their chemotherapy was, how far out after transplant they were. So there's a lot of variables here, but it's probably about 1% of the cells in the brain actually are from the donor after transplant. And there's things that can influence that a bit, but I think we're just on the cusp of being able to effectively treat neurologic disease, because we don't have a great way yet of delivering 3, 5, 10 times more cells into the brain. So if you're treating a disease that needs just a little bit of enzyme, then transplant may work well for that. And if you need to have more enzyme, then it may not work as well in terms of the neurologic aspects of the disease. So for instance, for Hurler syndrome, you may only need 1 or 2% of enzyme to make a difference in the brain. But for Hunter syndrome, you might need more because transplant doesn't work as well for Hunter. And for Sanfilippo, which is MPS 3, you probably need even more, and transplant doesn't really work very well for MPS 3 at all. So, again, if we had better ways of enhancing the number of cells delivered into the central nervous system, then I think we could have more effective therapy. But we're still trying to figure out how best to do that. You can look at those things in mice, because if you give more or less cells or different types of cells, or change the chemotherapy that you're giving, or whatever, you can then look at the mouse afterwards and see what proportion of the cells are donor cells. But it's a lot harder to do in the patients. You can't really tell by imaging. You can look at spinal fluid, but that's not really been all that helpful. There's hardly any cells in the spinal fluid to look at, so that's been challenging.
Yeah. Can you walk us through what a typical treatment looks like, the different stages in the process?
Well, so initially, we'd be interacting with the family about whether or not it might make sense to consider transplant for a particular child with whatever disease that they might have. One of the first things that we have to do is identify who's going to serve as a donor. And so in order to do that, we talk about HLA typing or tissue typing, and I kind of use those interchangeably. But all our cells have little markers on the cell surface that identifies those cells as being yours. And if you use cells from somebody that's not even closely matched, they're going to get rejected, generally speaking. So we have to establish the HLA type or tissue type on the kids that are getting transplanted. That usually takes about a week or so. That's done on a genetic level, actually. And then once we have that in hand, then we can do the search. So in the National Marrow Donor Program, which is actually here in Minneapolis, but those guys have these databases of both the cord bloods and the unrelated donors. And so we essentially just plug the HLA types into, well, they do it for us, but they will plug the HLA types into their database and give us a report about what kind of unrelated donors might be available that are closely matched. And the same thing with the cord blood side. So then we have to kind of decide, which donor are we going to use. Then we would bring the patients in and do, as we were alluding to before, assess the kids, see how they're doing. There's a lot of blood work that kind of goes into that, looking at kidney function, cardiac function, liver function, all those other types of things. And just to get a sense of what their health really kind of looks like. Part of that is what viruses had they been exposed to in the past? Because as you knock out the immune system going through the transplant, some of these viruses that may have been dormant for years can pop up and be a problem. So we want to know what issues we might have to deal with in terms of viruses as the kids are going through transplant. So that's about a week or 10 days generally to get all that information back. And then we would sit down with the family and kind of run through what we learned and then sign a consent. And essentially, here almost all the kids are treated on protocol. And so almost everybody gets the same package, so we can better understand how well it works, essentially. So the kids would then get admitted. They have to have a large kind of indwelling IV placed. And so that generally comes out of the chest. The tip goes into a big blood vessel by the heart, so we can draw blood whenever we need to. We don't have to stick them with needles. And so we can infuse whatever meds the kids might need. So there's two, we call ports on it, meaning you can be putting one thing in one side and another thing on the other side. So that is a surgical procedure. And the kids have to do that. We have to have that in place before we get started. So a lot of times we do that on the way into the hospital, essentially the day of admission, for instance. And then usually the chemotherapy starts. And the chemotherapy is all intravenous drugs. And we use actually antibodies too. So there's antibodies available to help knock out different parts of the immune system. So we don't have to rely on the chemotherapy to do all of that. And so that process, our current protocol is 9 days worth of the combination of antibodies and chemotherapy. And then generally there's like a day where you're getting anything to clear everything out of your body, and then you get the cells. So the unrelated donor cells show up fresh, essentially. So they've never been frozen. The cord bloods have all been frozen for some span of time. Those get shipped here, and then they get thawed kind of right outside the room in the hallway.
And they
then
smell. They smell.
And then all the cells are just given intravenously. So they circulate through the bloodstream and they localize in the bone marrow, and then they start to grow. But it can take a couple of weeks before you actually see the donor cells starting to recover. So during that span of time, so there's globally 3 different big pieces of the blood system. So there's the red cells that carry the oxygen around and get rid of the CO2 and stuff. And then there's the platelets that help you not to bleed. And then there's the white blood cells. And there's a large number of different types of white blood cells. But we can transfuse platelets and we can transfuse red cells. The white blood cells have to kind of come back on their own. So if we knock out the kid being transplanted, we knock out their cells, and it takes a couple of weeks for the donor cells to get started, there's a span of time where you have absolutely no immune system. And that's one of the big risks of the transplant. So the kids are in their own rooms, their private rooms. The air is all filtered to try to keep, mold and those types of things out of the air. And we pretty much keep the kids in the rooms until they've recovered enough of their white cells down the road where we think it's safe for them to get out of the room.
And that's generally about a month, correct? They're in the hospital after transplantation or after chemo for about
Yeah, well, so yeah, it's about 10 days generally from mission to the infusion of the cells, and it's usually about a month. Well, that's close to the minimum. I mean, sometimes people get sick and are in the hospital for a long time, but I mean, it's usually a month or so to recover your cells sufficiently to be able to be discharged.
I think Ollie was a couple days before a month. But as you're talking about this, it brings back so many memories. And I remember the workup week, which was, so intense and there's so much going on. And how everyone was surprised because when you guys were checking if Oliver had ever been exposed to any viruses, he hadn't. And everyone was really surprised, like he had really been exposed to nothing. And then when he got to the place where he could, get the cells, like he was ready for the transplantation, everyone was like, it's going to smell like canned corn. And I thought, no, it's not going to. It's this teeny little bag. How is it going to smell? And it does. It smells just like corn. It's wild.
Yeah, that's the preservative. So when cells have been frozen, it's dimethyl sulfoxide, or DMSO is what they use, but it's about 5% DMSO, and that helps the cells in the freezing and thawing process, so the cells don't lyse when they're going through that. So, but yeah, it has got kind of a characteristic smell to it. Some people can't smell it at all, but sometimes you're just walking down the hall and you go, oh yeah, somebody just got their cells. So, but that's only in the situations where people are getting frozen cells. So the fresh cells don't do that.
Now, walk us through the 100 days of isolation. How strict do families have to be?
Well, so as people are going through the transplant piece, I mean, there's again, these are all private rooms. There's a place for one of the parents to sleep. It's not the Ritz, certainly. There's these kind of fold out couches that are, functional, but they're not all that comfortable, frankly. And, Laurel can.
They're not too bad. They're not too bad.
And a lot of the families are kind of trading off who's with the kids. So, that's kind of an ongoing thing. When the kids get to be about a month out or so, generally, they're feeling better because a lot of the kids are been pretty sick. They're not eating very well. Some of them are on IV nutrition or enteral nutrition, like being fed with the tube, essentially. But early going through transplant, pretty much everybody's on straight IV meds. But as we're looking at getting them out, we have to go through that process of switching over the meds from IV to oral so the kids can take it and make sure they're doing okay from that standpoint. So that's one of the things that needs to happen before the kids can get out. If they're having fevers, we're worried about potential infections, so they're stuck in the hospital until we get rid of the fevers. And then, nutritionally, like I say, we need to make sure that either the kids are eating okay or the tube feedings or the IV feedings are such that they're doing all right from that standpoint. And they get out, and generally people go to Ronald McDonald here, and then we see them all the time in clinic for a span of, through 100 days, essentially. We want to make sure that their blood counts are doing okay and it doesn't look like there's anything infectious going on. And we're looking for complications and all these other types of things. As the kids get a little further out, it may go down from 3 or 4 times a week in clinic to 2 times to maybe once a week, kind of close to the end. And then before they go home, we want to make sure that we've got whoever's going to be taking care of them at home is kind of up to speed with what the issues are and what the meds are and what to do if the kid has a fever or whatever. So those are the pieces we have to kind of get in place before we would be sending somebody home.
Yeah. And once they go home, what does the protocol look like? Is it similar to what you would do for a newborn baby you're bringing home from the hospital, or are you just, or is it more intense? Are you sanitizing all the countertops, making sure nobody's coming over? Isolating in a little bubble for, a couple months. What does that look like?
Yeah, it's tough. I mean, generally the kids are still on immune suppression. So the idea is you're putting in cells from somebody else, and even if they're well matched, there's the chance that there's going to be an immune-related issue. So you can reject cells from a donor, like you can reject a kidney or a liver or something. So that's one set of issues. We have to knock out the kid's immune system enough that it decreases the risk of that. The flip side of that, though, is what we call graft versus host disease, meaning that donor cells are now growing in the kid's body, and they can see the new body that they're in as being foreign and try to reject the kids kind of from the inside. And so graft versus graft means the donor cells versus the person being transplanted as the host. So we put the kids on immune suppression for months, essentially, to try to decrease the risks of those things happening. And then as you get further out and the cells become accustomed to the new body, essentially, then we wean the immune suppression off. So that's one kind of big piece, is where the kids are in the immune suppression. Does it look like there's any graft versus host disease related kind of issues and those types of things? But regardless, when the kids go home, their immune system is not up to speed. And so we try to minimize the risks that they're going to pick up something from somebody. So people, it's easier with the adults, as you might expect, because if somebody's got a runny nose, they're afraid they got a virus or something like that. We just say it's better if you aren't exposing the kids that have been transplanted to that. The harder part is like the siblings and others, because if they're going to school and everybody's coughing on everybody and stuff, then trying to minimize the risks that a sibling or a cousin or whoever might come by and give you influenza or RSV or something like that. Those are concerns. So, I mean, we can't totally put the kids in a bubble so everybody recognizes that that's an issue, but we just try to educate people as best we can what the risks look like.
Yeah. And then I've got one last question. How many children have you transplanted?
I have no idea. I mean, it's obviously a team thing. It's not just me. I don't know. I'd have to kind of do some mental math here and try to figure it out. We transplanted probably 250 hurler kids. So there's adrenoleukodystrophy is another disease that we see a lot of. We probably transplanted 150 or 200 of those kids. And then there's, some of these diseases that you guys are familiar with that are much more rare, that are, more isolated issues. So
here's a better question. What's the most rare disorder that you've transplanted?
Well, that's a good question too. So, beta mannosidosis is kind of up there. So it's probably going to be in that sort of ballpark, I think, the kind of 1 in a million sort of incidence disorders. So there's a lot of very rare diseases, obviously, but it's I think that's probably as close as any.
You've talked a little bit about other treatments for lysosomal disorders. So I was just hoping you could talk a little bit more about those, like enzyme replacement therapy, gene therapy, and how you've been involved in clinical trials for those.
Yeah. So as we mentioned, I mean, the enzyme therapies have been around for about 25 years or so. I mean, as a pharmaceutical sort of product. I mean, there were individual investigators that were working on specific enzymes that, that were kind of one-offs almost, but it's really been about 25 years or so since some of these enzyme therapies have become available. So the idea is that these enzymes, when you infuse them, the cells can pick that up out of the bloodstream and internalize that enzyme, and it goes to the lysosome and does its job there. But it doesn't stay there very long. And so most of the enzyme therapies that are available, you have to give every week. So the enzyme infusions are 3-4 hours, maybe a little bit longer. You get a high peak in the bloodstream, and then the cells kind of snap it up and it goes away. And again, in a week or so, you have to do that again. So a lot of the patients have, we were talking about the catheter, the IV thing that we use for transplant. There are variations on that. A portacath is actually one of those, which they implant under the skin. And so you can feel it, and there's a little kind of rubber-like diaphragm piece on the associated with this. And you put a needle. The needle has to go through the skin, but it goes into the through that diaphragm piece into the port, and then you can infuse things. So a lot of families end up having these types of things implanted, and so you don't have to hunt around for an IV in the hand or something every time you get enzyme therapy. But it's a long-term commitment, and those enzymes tend to be very expensive. We just had a meeting last week. We were talking about some of these things, but Mark Dant, who was involved with a generation of enzyme for Hurler syndrome, or MPS 1 more globally, his son Ryan has this issue, and he was calculating that Ryan's gotten about $16 million worth of enzyme over his lifespan. So several $100,000 per year for each patient. Some of the enzymes are more expensive than others, and obviously, if you're bigger, your dose is going to be higher, so it's going to end up being more expensive. So, I mean, the way that things have kind of tended to work is that if you have a disease, which is an enzyme deficiency, and it's not primarily a neurologic disease, a lot of the patients will go on enzyme and just stay on enzyme. So, for instance, Hurler syndrome is kind of one of these where the severe kids, the kids that have mutations such that they make almost no enzyme, get sicker earlier, and they get developmental delay, and they progressively get worse. So those kids get transplanted because we have the ability to provide some enzyme into the central nervous system with the transplant piece. The patients that don't have a severe disease don't tend to have the same kind of neurologic deterioration. And so those patients just go on enzyme and stay on enzyme. So that's a big fork in the road for that enzyme deficiency, is how severe are you? If you're quite severe, there's a transplant pathway. And if you are less severe, actually you go on enzyme and stay on enzyme. And a lot of the other diseases where there's no neurologic issues, people just are on enzyme. The decision to choose enzyme versus transplant, it gets kind of muddy, but I think primarily, at least in the US and in Western Europe, it's based on risk. So transplant, for instance, might actually work better for some of these diseases than enzyme does, but there's still a 10%, 15% mortality associated with taking somebody to transplant. And so the geneticists have tended to be, can you justify that risk if you don't have neurologic disease, or whether the patient should just go on enzyme and stay on enzyme? And so, I mean, that's the way a lot of these decisions are kind of made. There are some families that have come to us that have said, we can't imagine giving enzyme weekly for 30 years for my kid. And I understand that there's a risk that is inherent in transplant that isn't in enzyme replacement therapy, but we're interested in taking that risk so I don't have to consider giving enzyme every week for the rest of my child's life. So there are those kinds of considerations that people think about.
And not all these diseases have enzyme replacement, right? I mean, like right now, beta-manocytosis doesn't have an enzyme replacement.
Right.
Do you know, like, approximately how many lysosomal disorders have an enzyme replacement to even consider?
That's a good question. I want to say it's probably in the 20-ish probably ballpark, maybe a little bit more than that.
Yeah.
But yeah, I'd have to look into it, frankly. I mean, I primarily see the kids that are being considered for, cellular therapies, gene therapies, transplant. And so if Fabry or one of these other disorders where nobody uses transplant, I'm not involved in giving the enzyme therapy for those patients. Sure. Because their geneticists are doing that.
But hypothetically, let's say you had a patient who is presenting more severe, would having a bone marrow transplant in combination with ERT together give the patient a better outcome?
Well, that's a great question. So, again, my background is kind of on the oncology side. There's very few silver bullets in treating cancers. So almost all the patients get combination therapy where one chemotherapy works in this way, another chemotherapy works another way. And if you combine them in, various doses and various schedules, you're likely to get much better outcome than you could with a single one on its own. So I think the door is open to consider doing combination therapy on these types of things. Some of the families that have gone through transplant here for, and I keep talking about her a little bit, but we've got probably more data on that than anything. But a lot of those kids get diagnosed, they go on enzyme therapy while the transplant's getting set up. And then some families say, we're used to giving enzyme. If we think giving enzyme might give my child a chance at a better outcome, we're okay with staying on it for at least a while. So some of the families have certainly done that. We haven't studied it in a rigorous kind of way. We talked to one of the companies that make the enzyme to try to see if they'd be interested in doing that. And they initially were kind of interested and decided they didn't want to do that. So it hasn't been formally studied, but we were trying to look at some of these outcomes and, do you need to have less surgery down the road? Are your cognitive outcomes better? Is your growth better? Those types of things if you've been on combination therapy.
Yeah, that's what I was kind of thinking. Everything you said about the neurological aspect for some patients, if they could have an enzyme that got into the brain and the transplant, and have, the transplant and then something that's getting into their brain where maybe transplant, can't get enough to the brain.
Well, so that's one of the directions that the field is headed a bit, and so as we talked about, these enzymes are large molecules. They don't get across the blood-brain barrier very effectively. So there are second generation enzymes that are now becoming available. So essentially you take the gene for that enzyme and you manufacture it such that it's got an antibody or a piece of an antibody that will interact with the vasculature in the brain, so it gets transported into the brain better. And so those types of things are being studied. Hunter syndrome is actually kind of more common in Japan, and there was a company there that developed an enzyme that was like that to treat the patients with Hunter syndrome, and that is approved in Japan now. So that enzyme is being tested in the U.S. And so that company's called JCR. There's another company called Denali that's doing something that's kind of similar to help better penetrate into the central nervous system with enzyme. And so those things are, one direction that we may have some therapies that are going to be better to offer than what we've had historically. So that's certainly been of interest.
Yeah, definitely. And then one last thing, just for those out there that don't really understand the difference between gene therapy and, let's say, a bone marrow transplant, because that's stem cell transplantation. Could you just talk a little bit more about gene therapy and kind of the direction things are moving in as far as gene therapy goes?
Sure. So when we talk about gene therapy, there's a couple different directions that you can go. For some of the diseases, like sickle cell disease, there's one mutation that characterizes sickle cell. And so there are groups now that have, there's very sophisticated ways to getting into blood cells and correcting that mutation. And so that's one direction to go. For a lot of the diseases, though, there may be a ton of different mutations. And so it becomes a little bit more challenging to have to correct, if there's 100 mutations in a gene or more than that, to correct each one of those. So a lot of the gene therapies for these kinds of diseases is, rather than correcting the mutation that's existing in the patient's cells, is actually just replacing a normal copy of the gene. And there's two big directions that you can go there. So one is actually delivering, for instance, a virus into the patient, and that virus may go to the liver or potentially to the brain or whatever it's kind of designed to do. and carry a copy of the normal gene and express that within whatever the tissues are. So that's what we call in vivo, meaning, in that person's gene therapy. And there's adeno-associated virus, or AAV, has kind of been the mainstay in doing that. The other one, and the one that we've been probably most involved with, is a bit different. So it's an ex vivo gene therapy, meaning where the cells are being corrected outside the body in the laboratory. And so the way that happens is that rather than using somebody else's blood stem cells that are potentially making enzyme, you take the kid's own blood stem cells and you insert a normal copy of whatever that gene is that they're missing. And generally that's done with a virus. And then you give the kid back his corrected cells. So the process there essentially is you identify somebody that you think this would make sense to do. There's a process by which we release their blood stem cells out of the bone marrow into the blood, and then you can harvest them from the blood. It's called apheresis is the process there, but then you can collect the kid's own blood stem cells. They're shipped to the manufacturing site, and there the blood stem cells are kind of purified, and they're exposed to this virus that's got a normal copy of the gene. They're frozen down, and then there's this process of checking it out to make sure that it went the way that you want it to go, that the cells are transduced is the terminology. Cells are transduced and they're not infected and they're healthy and all that other sort of stuff. So that takes about a month or 6 weeks to get that turnaround. Then we bring the kids back, and they still have to get chemotherapy because we have to get rid of their blood cells that haven't been corrected, the ones that are still deficient in enzyme. And then you infuse their corrected cells. And again, it's the same kind of thing. These cells are now frozen, but they're thawed outside the room and given in the IV, and they circulate through the body, go to the bone marrow, and start to grow. So from there, the process is fairly similar in that there'll be a span of time where they don't have any cells. It takes a few weeks for the cells to recover. Now they're your own cells rather than somebody else's. So we don't have to worry about the rejection part. We don't have to worry about graft versus host disease because the immune system is all the same. Kids don't have to be on immune suppression or any of those things. So it's safer. There's less complications associated with doing that. So as we were talking about the mortality, the chance of dying going through a bone marrow transplant, a standard transplant, maybe 10 or 15%. Probably at minimum, it can go up from there, but it's probably in the 1% ballpark.
Oh, wow.
If you're getting your own cells back. So it's,
That's
tenfold
huge.
safer, probably at least. So the other piece is, so there's two big considerations, right? One is the safety side, and the other is how well does it work? And so you can't get the normal copy of the gene into every last cell. So some of the cells are not going to be corrected. But in designing the virus, you can make it such that the enzyme that you're deficient with is expressed much higher than you would in a normal cell. So, you can look at, and it depends on which disease we're talking about, which virus we're looking at. But, one of our patients, I just got some of this data back last night, but one of our patients that have one of these leukodystrophy enzyme deficiencies, metachromatic leukodystrophy in this situation, but he's making probably 8-fold more enzyme than normal. And so you can potentially get better correction actually of the disease. So again, if you're only getting a small proportion of the cells into the brain, for instance, but now you're making 5 or 10-fold more enzyme, you can potentially get better correction. So that's kind of one of the things that we're very interested in being able to do and to look at, because it may end up being not only safer but more efficacious than what we used to do with transplant.
So would you ever then have, a patient that, let's say, had a transplant 10, 20 years ago go through this newer process?
Well, it's theoretically possible. So the way these things tend to work is that there's clinical trials that are done. The companies that are sponsoring these things work with the FDA and decide what patients they're going to look at in terms of the clinical trial. And so if it proves to be efficacious and the FDA says, yeah, it makes sense to actually license this as a product, then these are the patients that we are licensing it for. So they may say, patients with the more severe form of the disease or of a particular age range or whatever. And these gene therapies tend to be really expensive, like multi-million-dollar cell products. So whether the FDA would license that or allow us to use it in somebody that's already gone through a blood stem cell transplant, because that was not the population that we looked at in the clinical trial.
Sure.
So that's where that stuff is going to get kind of sticky.
Right.
So I don't know, if down the road you'd be able to do that, because I think, if the insurance is going to have to pay $3 million for a cell product in a patient we have no data in regard to their situation and using gene therapy in that mode, I don't know how well that's going to work, but theoretically it could be done.
Could you, so if a patient has had bone marrow transplant or enzyme replacement therapy previously, could that disqualify them for gene therapy? Is that what you're saying?
Yeah. So in the clinical trials, generally, yeah, the patients who have previously undergone transplant are not eligible for those clinical trials, because it kind of muddies the water, right? So, if you're looking at outcomes of patients that have previously been untreated and comparing them to patients that have gone through transplant before, then it's the data is less clean. You know what I mean? So
yeah, it'd be interesting to see, though, if they would ever approve someone who wasn't faring well, who was deteriorating, and approve them for it.
Well, I mean, I think one can envision that there may be a possibility to do that. There's actually another gene therapy that we're looking at here that's a bit different than looking at the blood stem cells. So in that situation, we're looking at engineering B cells. So B cells are the ones that make antibody, and they can churn out a ton of antibody. And so there's a company that we're working with that where we're just isolating the B cells from a patient, and it's a different delivery system. This is actually a transposon-based delivery system as opposed to a virus. And I don't know if we need to go into all the details there, but essentially you're just engineering the B cell population and not the blood stem cells. And so in that situation, you aren't actually giving chemotherapy. You're just infusing the patient's own cells right back to them. So yeah, I mean, maybe something like that could be done. So if somebody's gone through bone marrow transplant and the proportion of cells that are from the donor are fading out over time, which happens infrequently but can happen. So if you look in the blood, maybe you had 80% donor cells there, and then later it's 60 and 40 and 20. So the benefit that you're getting out of your transplant is deteriorating as you're losing donor cells. Maybe in a situation like that, you can rationalize doing a gene therapy for what, patient. So I don't know. I mean, I think those kinds of things are going to come up down the road, but it's pretty early to be able to say how successful we're going to be in being able to offer those kinds of therapies in situations like that.
It's exciting though that we're even talking about it, I think,
Yeah, it really is. I mean, I think there's for years we've really not had anything new to offer the families, and we've been tweaking the transplant piece, trying to make it a little bit safer kind of around the edges. But really, if somebody was transplanted 30 years ago and they got all donor cells, that's all we can achieve now with transplant. It's safer potentially, but it's not any more efficacious than what we were doing years ago. So having the opportunity to try some of these other therapies, the gene therapies and the new enzyme therapies and whatnot, I think is really exciting, because these things may prove to be better, and we don't have to deal with the complications that are inherent in the transplant process.
Yeah. What do you think is the most exciting area of research in the field of rare disease right now?
Well, I think there's a couple pieces. In the first place, we're better understanding the biology of these diseases, and because it's not just the accumulation of whatever material the enzyme can't break down, but for instance, there's emerging appreciation of inflammation and other types of things that are associated with this that might be really fruitful in terms of additional therapies. But from my standpoint, the kind of things we're doing here, having the opportunity to offer these gene therapies and to really determine how effective they are and how we might best utilize them is really an exciting thing.
Yeah. So if you could see one major breakthrough in the next 5 years, what would it be?
Well, so I think one of the big challenges that we're spending a lot of time talking about is how can we do the financial aspect of some of these things? These rare disease interventions and gene therapies, for instance, are multi-million-dollar projects. I mean, some of these companies are spending hundreds of millions of dollars doing clinical trials, and that's just not viable for some of the ultra rare disorders. Whether or not the FDA will develop different pathways so that, for instance, those of us in academia can potentially develop our own vector systems and try those without having to necessarily partner with industry. And, like I say, I don't know if the industry is going to even be involved in some of those things, because it's really difficult to understand how they're going to make money doing that. So, are there new paradigms that we can put together that is going to allow us to better intervene for some of these very rare disorders is something I'd like to understand better.
Dr. Orchard, what keeps you hopeful and energized in this work after all these years?
Well, I get to see a lot of patients back that I treated a long time ago, and some of those have graduated from high school. Some even have started their own families. So that's really something that's exciting to see. It makes me feel old sometimes, but that's okay. There are kids that we're seeing that are relatively young yet that are making much more enzyme than they would otherwise be making, and we're very looking forward to see what their lives are going to look like. So that's something that kind of keeps me coming to work as well.
Yeah, that's incredible.
As parents and advocates, we often hear the phrase, "These kids can't wait." In fact, it's why we adopted the tagline to the podcast. What message would you share with families who are still awaiting some of these answers or options?
Well, I think the role of families and advocacy groups and helping move the field forward really is important to understand. And you know, talking to Congress people or senators, anyone who will listen about the unmet need and how we can move these things forward is huge. So the families have a lot of power in this, in terms of telling their stories and helping people understand what the need is. And don't be shy about talking to people about this, because that can make a huge difference.
Dr. Orchard, thank you so much for joining us today and for sharing your incredible work and insight. Your dedication reminds us of why the world of rare disease need more voices, more research, more urgency, because these kids can't wait. If you've enjoyed today's episode, please subscribe, leave a review, and share. You can also visit our website to learn more about the Lost Enzyme Project and make a donation to important research being done right now. All links will be provided in the show notes below. Thanks.
Thank you.
Thank you for joining us on These Kids Can't Wait. Every story shared brings us one step closer to awareness, advocacy, and action. If you've been inspired, please subscribe, leave a review, and share this episode.
To learn more about the Lost Enzyme Project or donate to the cause, you can visit our website, Instagram, or Facebook page. Those links will be provided in the show notes below. Thank you again, and remember, these kids can't wait, but together, we can make a difference.